So we'll do the same again. We'll take questions at the panel. So, well, no, we're going to do them at the panel. The panel. So the next speaker is Brandon Dekosky. Oh, Brandon, sorry, I can't see you for looking at you. Okay, go ahead, Brandon. Yes. So Brandon is from the Reagan Institute at Mass General, MIT, Harvard, and he's an associate professor, and he's going to talk about antibody functional screening against challenging drug targets. Go, Brandon.
All right. Well, thank you so much, Dennis, and Fair Journey and the conference organizers. It's such a pleasure to be here and share these couple days with you all. Really exciting talks, and excited to follow all the speakers. And you'll see I'm going to talk about some HIV, so it's really exciting to follow Gary as well in that area. So just some quick notes about high-throughput adaptive immunity and where we start. Of course, everyone in the room is familiar with the heavy and light chain pairing problem. Antibodies are a heterodimer of both heavy and light chains. And when you're doing discovery, you need to find a way to pair them in some way. This talk is not about T cells, but a lot of the technologies just going to point out really apply to T cell receptors as well. Maybe even to a higher degree. This is a crystal structure, and you can see how the peptides at the center of the CDR3s make interaction with the peptide bound in the MHC groove shown here, and then interactions outside. And so both of the immune receptors have the same potential sticking points in terms of discovery. Getting the natively paired heavy and light chains is really important because it allows often better developability properties and often superior neutralization. If you're looking at viruses, maybe superior performance. So it's really valuable to be able to capture those. And that's one of the places that our lab has really worked in for a long time. So starting with my PhD work at University of Texas and through my postdoc at VRC and now after some number of years of having our lab, we've really been developing a lot of approaches that try to blend the best advantages of both single-cell technology, but also high-throughput screening and high-throughput sequencing. And in particular, we're very interested not just in getting paired sequences, but also in finding ways to screen those paired immune libraries for function and start to map function onto the large sequence data that we're able to collect. And so this is the general approach that we've settled on over the years. It works quite well for recovering antibodies, and most recently, we've also published it for T cells as well. The general idea is here in panel A, we isolate single cells into droplets, and then inside those droplets, we capture mRNA onto magnetic beads. So then we can wash away all of the cell lysate and all of the things that really make RT-PCR reactions inhibited in some ways, and then re-emulsify each of those beads and do an emulsion linkage RT-PCR. So that actually physically links the heavy and light chain genes onto one strand, and once they're physically linked, we can sequence them, and we can also clone them, as we'll get into in a couple of slides. And so, again, we've gone through a few variations of this over the years. The droplet-based approach seems to work quite well, and we've used a number of different sequencing platforms, the MySeq, the PacBio, things like that. And so using this approach, it's fairly simple in our group to capture 100,000 or more unique heavy and light chain pairs. So that's clonotypes. That's not like the number of cells. That's the number of unique clonotypes that we see in a highly validated, rigorous bioinformatic analysis where we clean out all of the potential errors. And the same thing from T cell receptors, which we just had a couple of publications in over the last year or so, where we're recovering in the mid-tens of thousands of unique T cell receptor alpha and beta chains. Of course, we're all here about antibodies. Antibodies are a bit more diverse because of somatic hypermutation, but T cells are also quite diverse. So once we have those paired heavy and light chain genes, the next thing that we really started to look at doing is how do we start mapping these for function? And in particular, we were looking for a way that you could do repeated screening, so capturing as libraries, and then enabling screening against different antigens, different epitopes. You know, sometimes you don't know exactly what cross-reactivities you're looking for. Maybe you want cross-reactivity to rhesus, mouse, human, things like that. And so it's really helpful to be able to interrogate that library repeatedly. And we found that yeast display was a really effective solution for that. So what we did was we added restriction sites into the linker region at the center. And then, of course, it's trivial to add them to the outside just by doing another round of PCR. And enabled cloning into a yeast display vector. So we've got a bidirectional promoter here based on it's galactose inducible. So when you feed the yeast galactose sugar, they'll start kicking up translation of this particular gene. And added a number of different kind of components to make sure that we were getting really high fidelity expression of a representative panel of human antibodies. Because, of course, if your library is diverse, you need rigorous ways to display it as well. And so this is the construct that we ended up using. We use a Fab format. Again, there's lots of great applications of display. There's different formats, Fab, SCFV, things like that. And of course, there's yeast, bacteria, phage, and everything is good for you know, what it is. And this is just the platform that we chose for a few different reasons. And so over the years, we've published this in a number of different settings. In the first publication, we found a panel of broadly neutralizing antibodies against the HIV fusion peptide. I'll actually talk a little bit more about that specific problem later. Things like mapping to different donors and different variants of viruses. So this was with flaviviruses, so looking at cross-reactivity as well as the maturation state of the virus. So there's the immature form and the mature form of flavies, and both coexist within an individual during an infection. So which antibodies are targeting which kind of circulating variants of the virus or stages of that viral life cycle. And during the pandemic, did some work in trying to understand what is it that leads to the best potency against SARS-CoV-2 for a particular epitope, and how does that work? So just the most recent study that we've done with functional screening, this was actually a collaboration with partners at Sanofi. And we mouse immunized with a model immunogen, and we captured immune libraries, natively paired immune libraries from bone marrow, spleen, from bone marrow and spleen. And in particular, we were excited about analyzing the plasma cell population in bone marrow because it doesn't express surface B-cell receptors. And so you can't screen it using a traditional antigen bait approach. And so by capturing those paired heavy and light chain genes, cloning them into display, we were able, after just a single round of screening, show that the library was extremely enriched. So on the y-axis, you're looking at antibody expression in Fab expression on the yeast. And on the x-axis, you're looking at antigen binding. And you can see, 35-fold enrichment across one round. It's just, really exciting to see. Similar thing from spleen cells, and part of this that we haven't published yet, we're still working on, is we actually scrambled the heavy and light chains, and we're looking to see how does that scrambling of heavy and lights impact affinity, what pairs do you get out, and what are their properties. So we've been exploring different ways to try to map at a coarse grain level the affinity of these populations. So for example, taking plasma cell populations and cloning them, screening them, mapping for affinity. And as you can see, on average, it works pretty well. It could use some improvement, obviously, refining the algorithms based on these data of how can you really mine yeast display data to get better affinity predictions even before you've made a soluble molecule. In red, those are those antibodies that had a really high K-off, and I'll just point out that some of the antibodies that were discovered using this approach were just as good in terms of affinity as the best that our partners at Sanofi had against this target. So that was really exciting to see that we were able to get those kinds of really, really powerful antibodies. So building on the discovery, the natively paired discovery platforms we've been using, we started to think, okay, sometimes the antibodies that we're able to isolate from an immunized mouse or animal need some engineering, as you all know. And how can we learn about, using these high throughput approaches, how can we engineer better antibodies? And so in particular, we've been focused on the fusion peptide epitope of HIV. So as Gary had mentioned, there's all of these different vulnerable epitopes on the HIV surface. Often the virus has a consequence to mutating these sites, and so they're more broadly conserved and also vulnerable to neutralization. The fusion peptide is down here, and it's a really promising vaccine target because that fusion peptide site is short, 8 amino acids, and so it doesn't vary. I mean, it does vary, but it's a little bit easier to target with a vaccine. But the major issue with fusion peptide is that the antibodies against that site are less potent and less broad in general than the other sites. So of all of those really exciting antibody specificities that have gone into clinical trials and are starting to show some promise in the field, none of them have been targeting the fusion peptide. And we wanted to know why and what could we do about that. And so a postdoc in the group, Bharat Madan, started to really take this on. And we started to use an approach called site saturation mutagenesis to make every possible mutation at every location and screen for affinity. Of course, using next generation sequencing, we could map the affinity of each of those mutations and try to identify what is really going to push affinity and neutralization against this strain. So this is just a heat map. The y-axis shows all the different possible amino acids at each site, and the x-axis shows the position along, this is just a template numbering scheme, so the position along the gene. The white circles are actually the native form of that particular antibody, and you can see that only around 1% of mutations will enhance affinity, versus around 40% of mutations are deleterious. They're really going to end up knocking down affinity in one way. And so you can imagine the advantages of adding one mutation at a time, as we're doing here, and trying to identify what are the best mutations and bringing them along for better development. We did some structural analysis. This particular antibody had an interesting mutation that kind of bridged interactions between heavy and light chain, and actually changes some of the dynamics at the VHVL interface, and that was what led to better improvement of this particular molecule. But, you know, kind of just to cut that story short, single mutations were really not enough. They were giving several-fold improvement in potency, but it wasn't enough for this particular vaccine, for this particular HIV epitope. And so building there, we started with a human-derived antibody, and we started to do iterative screening. So taking those first round of mutations, the single mutations, and combining them with DNA shuffling heavy lights, reselecting to try to see what is it that can give us better recognition of this site. In particular, there's one fusion peptide variant that has an insertion that was really a problem for recognition. So this is a fairly prevalent sequence in the global HIV representative strains, and this particular antibody wasn't able to recognize that at all. And so after a first round of screening, we started to really incorporate that into our screening rounds. And just a note, we're screening the trimer against the BG505, so this is a stabilized version of the HIV trimer, where we swapped different variants of just that 8 amino acid fusion peptide sequence. So these are some examples of the yeast display plots, and I just don't want to dwell on them too much, but I just want to show when you have the single mutations with some of the libraries against some of the antigens. So this is the heavy chain library and the light chain library, and then you've got 3 different fusion peptide antigens on the x-axis. Some of these, you see some substantial improvement. In red is the improved library versus blue is the template. But in others, you know, maybe it didn't improve quite as well. When we started combining heavies and lights together and we started shuffling mutations, then we were able to get a much broader representative improvement of recognition across the different diverse panels of the HIV FP. And in particular, this is that V4 variant with the insertion, and you can see that it's been really dramatically enriched. The concentration here is over 20-fold higher than that kind of standard version that it's neutralized. The wild type doesn't bind to it at all, and then this mutated variant binds to it very nicely. So just to kind of skip to the functional data, so first we set up a 20-virus panel because there's so many strains of HIV and you want to down-select your candidates. And we wanted to pick based on something that, an optimized panel that was going to be representative of antibodies that target this site or this interface. And this particular variant neutralized 100% of those strains in the panel, and it turned out after, after one pandemic of waiting to get the neutralization assays back up and running, we ended up being right on the projection, which was amazing. I've never seen extrapolation like this before, and I don't expect to see it again, but it was really exciting to see that pan out. So this particular antibody starts to neutralize a much broader swath and more potently of circulating HIV variants, which is actually puts it as the most broad and potent antibody to directly recognize that fusion peptide. And so if you put it on a plot, this is actually very similar to a plot that you saw in the previous talk. It brings this fusion peptide antibody from outside the range of antibodies that have come to the clinic to being right there in some of the first generation clinical antibodies. So really exciting to kind of bring this epitope into that range. So structurally, the way it works is there's actually 3 substitutions. There's an E2K mutation, which is actually surprising. We wouldn't have thought that one of the best mutations was going to come at the second residue of the heavy chain. And it actually forms kind of a second bridge with the E87 of the HIV trimer. There's a more kind of traditional substitution that is interacting directly with the fusion peptide here, this G514 of the fusion peptide. And then the last was another really interesting one. It was an A33P mutation, and that actually makes space in the binding groove, so the fusion peptide in purple is shown here. And it was that A33P mutation that enabled us to pick up that insertion in the fusion peptide. It broadens that binding groove and gives space for broader recognition. So we're really excited about this to kind of demonstrate that the fusion peptide can be brought into the clinically relevant range and bring in a new epitope target against a very difficult virus. So just a quick word on applying some of these screening strategies for malaria. This work has been published, so I don't want to spend too much time on it, but just a note that malaria has nearly 500,000 deaths per year. The vast majority in children, and low-cost solutions are really urgently needed. Colleagues in the Bob Cedar Group and VRC, as well as many other groups around the world, have started to really identify antibodies as one way to use antibody prophylaxis to prevent new infections. And in particular, the sporozoite is a great target. We heard about the blood stage targets this morning, which are awesome. And then these are the sporozoites. So this is the stage of malaria that is injected by the mosquito and traffics to the liver. So that's another opportunity to interrupt the life cycle. And in early trials, some large doses showed that you could have substantial protection, and that actually did extend to the field. And so the question is, how can we improve the potency of these antibodies and get them to the range where they would be relevant in the field? So just a quick note about this particular target. It's a very tricky target because the circumsporozoite protein that covers that sporozoite has a lot of repeats, and it has no known structure. It's got a lot of disorder. And so in blue, these are all of these different NANP peptide repeats. And this particular antibody recognizes this peptide shown in green, sometimes called the junctional epitope of NPDP. And so it's a very similar structure, and you could imagine trying to get things that recognize this peptide versus all the repeats. The issue that makes it very complicated is that this antibody binds to both. So it binds to the repeat peptides at a somewhat lower affinity, and then the junctional peptide at a higher affinity, and that seems to be how it gets avidity while recognizing just one unique peptide that is the protective epitope. To make a long story short, and again, this one is published so you can read about it, it's a little bit complicated. There's no neutralization assay, so you have to essentially try out promising antibodies in mice to find out if they're protective. And it turns out that one of these indeed was protective, and it recognizes this junctional epitope, peptide 21, to a substantially higher degree. So we're really excited to be continuing in this work of generating improved anti-malarial therapeutics that could be useful for broad, long-term protection campaigns. So the last thing I want to talk about is some new functional screening capabilities that we have been looking at. Everything I've talked about before has been affinity-based, which is awesome for some targets, but sometimes you're looking for more than just affinity. So virus neutralization is a really good example of that. So if you're doing a virus neutralization assay, it's an analytical assay, so you've got all these well plates that you're screening, and each one has a lot of cells in it. And it's a problem because it's really an analytical technique. It's not as useful for evolution or selection. You make every antibody one by one, and then you find out which one works. And the antigen proxies, even when they're available, they're often poor substitutes. So, just because you have binding to a BG505 SOSIP or a SARS-CoV-2 fusion protein, it doesn't mean that that antibody neutralizes virus. And so it makes it hard to select for the property we really want, which is neutralization, when all we can do is select for binding. And that's a really big gap. And so what we've been trying to do is take these 10,000 cells per well screens, where that's one assay, and actually move that into droplets. So each droplet is one assay, and then you can actually take 10 to the 6 assays and move them into just a single microtube, and use a computational NGS readout so that you can identify what are those most neutralizing antibodies directly, and use directed evolution to try to select them better. You can add mutations across rounds, things like that. And so this is the way it works with an HIV model. So what we've done is we take a TZM GFP cell, which is a variant of the most common cell line that's used for neutralization luciferase-based, which is TZM BL. And this variant was developed that expresses a fluorescent molecule on infection. And so we make this this cell is both capable of infection, and we can engineer it to secrete an antibody. And so we can have a library of these cells. Each one of them is capable of infection, and each one secretes its own antibody. So when you put that cell into a droplet, it will secrete into the droplet and start to form its shell of potentially protective antibody inside that droplet. And so just like a well plate assay, first you add the antibody, and then you challenge with the virus. And so in this case, we merge that droplet containing antibody with another droplet that contains an HIV pseudovirus. And inside the droplet, that challenge happens. And two things could occur. If the cell is secreting a neutralizing antibody, it will remain dark. It's not going to express that signal. And if it's a non-neutralizing antibody, so it doesn't have the function we're looking for, it will start to express a GFP. So the nice thing about this is that we can now recover the cells directly. We just get them back from the emulsion, and we can use flow cytometry to sort at a very, very fast scale. So it's very simple. You just sort the GFP positive versus the GFP negative cells, and using next generation sequencing, you can identify the antibodies you're after. Depending on how the assay is set up, you might be able to recover these dark cells, add mutations, do another round of screening for directed evolution to evolve directly on that neutralization property. So this is the way it works with a synthetic library. So the vast majority of this library in black is a non-neutralizing antibody called 72A1. And then there's two neutralizing clones in here. One is VRC01 and one is VRC34. Both neutralize the virus strain that we're working with. So if you take the library of cells, this synthetic library, put it into droplets, merge the droplets, do the droplet challenge, get the droplets back, and then sort, which sounded like a long run-on sentence, but it really only takes a few days once the library is made. Then you look at the non-infected population. Well, with the HIV model, you don't get a lot of infection just out the gate. Even in well plates, you'll get 5-10% of the antibodies are infected. And so you don't see a lot of change in this population with this particular model. But if you look at the GFP positive cells, you'll see that they're actually completely depleted of the neutralizing antibodies. They're all gone. So the cells secreting neutralizers very efficiently protected against that challenge in droplets. And actually, we can do sequencing and estimate and look at the numbers. So essentially using the same approach as in yeast display, where we look at enrichment of different clones across rounds. In this case, we really just needed one round, and we're seeing 350 to 1,000-fold selection in just a single round of sorts. So very efficient, very strong signal. And we've got ongoing expression from a number of libraries and a number of different targets, not just viral, but we're also looking at other targets with this type of approach. And so we're really doing a lot of functional screening in droplets these days. We're doing several million neutralization assays a week using these droplet approaches with a very small team working on it. We're doing direct neutralization. It works with natively paired libraries and also with synthetic libraries. It's compatible with different formats, multi-specifics, things like that, fusion proteins. And one of the things we're most excited about is, again, we don't need those extracellular domains to be produced and purified or imperfect model or surrogate antigens. We can just do this with the pseudovirus or the virus directly. Another thing that we're excited about is, of course, broad viral panels. You could imagine using a broad panel and rapidly screening for breadth. And again, we're going into additional challenging targets with a particular focus on GPCRs, and hope to talk about that soon as well. So with that, I just want to thank all the folks who have really been working together with us in putting everything together. Of course, at my PhD lab, George Georgiou at Austin, where the original paired heavy and light chain sequencing platforms were developed and evolved. And my postdoc, John Mascola, and many others at Vaccine Research Center, some of whom are continued collaborators, including Peter Kwong, Bob Cedar, Danny Duick, and of course the group at University of Kansas first and now at MIT. So it shared a lot of work today from Bharat Madan, Bailey Bonek, Ahmed Fahad, Xiaoli Pan, Matias Gutierrez, and Mateus Oliveira de Souza. And with that, thanks again. It's wonderful to be here and looking forward to the panel.